Hello students, welcome to the first lecture of the 4 lectures on glycogen, glycosaminoglycans and glycoconjugates. Following these 4 lectures, you should be able to discuss the structural aspects of glycogen and the key bonds involved in its formation, the basic aspects of glycosaminoglycans, or as they are abbreviated as GAG. You should be able to discuss the clinically relevant glycosaminoglycans. You should be able to define the different classes of glycoconjugates, which we shall look over these 4 lectures. You should be able to discuss the specific structural characteristics and functions of proteoglycans and glycoproteins. You should be able to relate the different clinical aspects of mucopolysaccharidosis or MPS and eye cell disease. And in this particular series of lectures, we will be talking about blood grouping, but we will not be focusing on the details of this particular aspect, but you can read through the slide that I have provided where this particular topic has been discussed and elaborated in detail, and we can look at this in a revision session prior to the examination. One point of note, you do not need to focus on the chemistry of the pathways for the synthesis and degradation of glycoconjugates because they are not important for your boards or assessment. So that's actually the summary of what we will be covering over the next 4 short lectures. So let us start with the structural aspects of glycogen and the key bonds involved in its formation. What are the basic facts about glycogen? And some of these facts we have discussed in detail when we took carbohydrates, or more precisely, introduction to carbohydrates. Glycogen is a polymer of glucose, and this fact is already known to you. In humans, it is deposited in the liver and muscle as storage material, which are readily available as immediate source of energy during times of need. And this particular fact is also known to you. Shown here in the slide is the biopsy sample from the liver of a well-fed mice. And you can see here the hepatocytes show large deposits of glycogen. This particular part of the slide shows you the biopsy sample from a mice which has been starved for a period of 24 hours. And you can see here that there has been significant depletion in the glycogen stores because glycogen in this particular animal has been broken down by the process of glycogenolysis to produce glucose to sustain the different energy needs of the body. We also know that formation of glycogen from glucose happens through a process known as glycogenesis, and we shall look at this particular metabolic pathway in detail later in the course. We also know that breakdown of glycogen to form glucose is known as glycogenolysis, and this particular metabolic process we will also study in detail later in the biochemistry course. The process of glycogenolysis is affected by pH, and you can see here from the information provided in this part of the slide that post-mortem glycogenolysis is very rapid but ceases when the pH falls to 5.5 because of accumulation of lactic acid, or lactic acidosis leads to the inhibition of the process of glycogenolysis. So this is a take-home message for you which you should remember. Before we go into the bonds that are involved in the formation of glycogen, let us go back to a very basic concept in organic chemistry known as the formation of hemiacetal. When we have an aldehyde group and when we have an alcohol group, under certain conditions the alcohol oxygen adds to the carbonyl carbon, leading to the formation of hemiacetal, and this happens through this particular intermediate. If you do not remember the intermediate, just remember that a hemiacetal is formed when an alcohol oxygen atom adds to the carbonyl carbon of an aldehyde or ketone. And this particular aspect is important, as in the next slides we will discuss about the cyclic structure of glucose. So this is the linear structure of glucose. As you can see here, glucose has an aldehyde functional group. It also has hydroxyl groups. Under certain conditions, they can undergo an intramolecular reaction to form a cyclic hemiacetal or hemiketal. And this particular aspect is shown here in this part of the slide, where you can see here the oxygen reacts with the carbonyl carbon and forms a cyclic structure because of the formation of hemiacetal. And there is an intermediate involved, but we do not really need to know this particular aspect, but we need to look at this particular part of the slide where the cyclic structure is formed because of the formation of a cyclic hemiacetal. Based on the position of the hydroxyl group, the hemiacetal or the cyclic structure of glucose can have two conformations. One is called the beta anomer, when the CH2OH and the hydroxyl are on the same side. Similarly, it can have an alpha anomeric conformation when the hydroxyl group and the CH2OH groups are on the opposite side. So, depending on the position of the CH2OH and the hydroxyl group in the cyclic structure of glucose, we classify them into two beta anomeric form when the CH2OH and the hydroxyl groups are on the same side, and the alpha anomeric form when the CH2OH and the OH are on the different sides. By now, Prof. Riyad would have taught you about ether linkages. So this is the ether linkage that is present in the cyclic structure of glucose. Now, if I want to number the carbons in the cyclic structure, how will I do so? I look at the ether linkage and then I choose the carbon which is nearest to the ether linkage, which is this particular carbon. We label this carbon as 1 and then successively count the number of carbons from this particular carbon. So here this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, this is carbon 5, and this is carbon 6. Remember that the glucose molecule has 6 carbons because it's a hexose. Now, in case of glycogen, what is happening is that there is a linkage between the first and the fourth carbon. So, if you look at this particular part of the slide, this is the first carbon which is placed here, and this is the fourth carbon that is placed here, and there is a glycosidic linkage that is formed between the first and the fourth carbon, and this particular carbon is called, this particular linkage is called the alpha 1,4 glycosidic linkage. Why it is called the alpha 1,4 glycosidic linkage? Because of the involvement of the alpha anomeric form of glucose in this particular bonding. So in glycogen we have 1,4 glycosidic bonds that are formed between the hydroxyl oxygen atom on carbon 4, which is located here, and the alpha anomeric form of C1 on the other, which is located here. So in glycogen, the first kind of bond that we encounter is alpha 1,4 glycosidic bond. In the books, sometimes it is written as alpha 1,4 glycosidic linkage. And this particular carbon is the anomeric carbon because this is located near to the ether linkage and therefore is designated as the anomeric carbon. Similarly, the second kind of bond that we encounter in glycogen molecule is the alpha 1,6 glycosidic bond. Here we have the sixth carbon, and this is forming a glycosidic linkage with the carbon at position 1, that is the alpha anomeric carbon. So this is the ether linkage, this is position 1 of the first carbon, and there is a glycosidic linkage happening between the first and the sixth carbon. Therefore we call it an alpha 1,6 glycosidic bond. In other words, an alpha 1,6 glycosidic bond is a bond between the hydroxyl group on carbon 1 of one glucose and the hydroxyl group of carbon 6 of another glucose. So in a glycogen molecule, we will have two kinds of glycosidic linkages, alpha 1,4 glycosidic linkage that we looked at in the previous slide, and alpha 1,6 glycosidic linkage that is shown here in this particular part of the slide. Also, when we study the metabolism of carbohydrates, especially glycogen, you will encounter a term called the reducing end. The reducing end, to be precise, is the end where the anomeric carbon is located. How do you identify the anomeric carbon? Very simply, the carbon that is located next to the ether linkage is called the anomeric carbon. And this is also the first carbon, and we label the successive carbons taking this carbon as a point of reference. So in this case, this will be 1, 2, 3, 4, 5, and 6. And you can see here, these are the alpha 1,4 glycosidic linkage, and these are the alpha 1,6 glycosidic linkage. Now, if I look at the structure of glycogen quickly, then I can see that there are two types of linkages that are existing, the alpha 1,4 glycosidic linkage and the alpha 1,6 glycosidic linkage. Presence of these two linkages helps in the branching of glycogen, especially the formation of the 1 alpha 1,6 glycosidic linkage, and by branching the glycogen molecule allows the storage of large number of glucose molecules in a limited space. As I said, we will talk about the process of making or synthesis of glycogen or the breakdown process in separate lectures during the later part of the course. But at this point of time, I believe you should be able to understand what are alpha 1,4 and alpha 1,6 glycosidic linkage, as well as the cyclic structure of glucose, which is formed because of the formation of an hemiacetal. Also, you should be able to identify what are the alpha and beta anomeric forms of glucose. So that brings me to the end of this first lecture. I believe if you have any kind of problems or doubts, please let me know and I will be able to address it. Thank you for listening and have a very nice day.